All living organisms are made up of cells, but not all cells are the same. Scientists classify cells into two main types, prokaryotic and eukaryotic, based on their structure and level of organization. Prokaryotic cells are simple, small cells that lack a true nucleus and membrane-bound organelles. Their DNA is found in a region called the nucleoid, which is not enclosed by a membrane. These cells are typical of organisms such as bacteria and archaea. Eukaryotic cells, on the other hand, are larger and more complex. They contain a true nucleus that encloses their DNA, as well as membrane-bound organelles such as mitochondria, the endoplasmic reticulum, the Golgi apparatus, and, in plants, chloroplasts. All plants, animals, fungi, and protists are made up of eukaryotic cells. Despite their differences, prokaryotic and eukaryotic cells share several common features. Both types of cells have a cell membrane that controls the movement of substances in and out of the cell, cytoplasm where many chemical reactions occur, ribosomes for protein synthesis, and genetic material, DNA, that carries instructions for cell activities. These similarities show that both cell types share a common evolutionary ancestor and perform the same essential functions needed for life. However, there are many structural and functional differences between the two. Prokaryotic cells are much smaller, usually between 0.1 and 5 micrometers, while eukaryotic cells range from 10 to 100 micrometers. Prokaryotes reproduce asexually by binary fission, a simple process of splitting into two cells, whereas eukaryotes divide by mitosis and produce gametes through meiosis, allowing for greater genetic variation. The DNA of prokaryotes is usually circular and not wrapped around proteins, whereas eukaryotic DNA is linear and associated with proteins called histones, forming chromosomes inside the nucleus. Eukaryotic cells have a well-developed cytoskeleton that gives them shape and allows organelles to move within the cell. This structure supports more complex functions, such as cell division and intracellular transport. Prokaryotic cells, while lacking membrane-bound organelles, sometimes have specialized structures like flagella for movement or plasmids for carrying extra genetic material. Their cell walls are made of peptidoglycan in bacteria, while eukaryotic plant cells have walls made of cellulose and fungi have walls made of chitin. The differences between prokaryotic and eukaryotic cells are extremely important. The complex compartmentalization in eukaryotic cells allows them to perform specialized tasks efficiently, which supports the development of multicellular organisms with specialized tissues and organs. Prokaryotes, being simpler, can reproduce quickly and adapt easily to changing environments, making them vital to ecosystems as decomposers and nitrogen fixers. The structural differences between these cell types are also medically important, as certain antibiotics can target bacterial cell walls or ribosomes without harming eukaryotic, human, cells. In summary, prokaryotic and eukaryotic cells differ mainly in complexity, organization, and internal structure. Prokaryotes represent simpler, single-celled organisms, while eukaryotes have evolved to form more advanced, multicellular life forms. Understanding these differences helps scientists explain how life is organized, how it functions, and how it has evolved over time. Cells are the building blocks of all living things, but not all cells are the same. Scientists classify cells based on their structure, complexity, and function. The most important distinction is between prokaryotic and eukaryotic cells. Prokaryotic cells are simple cells that do not have a nucleus or membrane-bound organelles. Their DNA floats freely in the cytoplasm. These cells make up unicellular organisms such as bacteria and archaea. Eukaryotic cells, on the other hand, are more complex and contain a true nucleus surrounded by a nuclear membrane. They also have organelles such as mitochondria, endoplasmic reticulum, and Golgi apparatus, which carry out specialized functions. Eukaryotic cells are found in plants, animals, fungi, and protists. The classification of cells became possible because of advances in microscopy. Early microscopes allowed scientists like Robert Hooke and Anton van Leeuwenhoek to observe cells for the first time. Hooke discovered and named cells when viewing cork slices in 1665, while Leeuwenhoek later observed living cells and microorganisms. As microscopes improved, scientists could see internal structures such as the nucleus, cell membrane, and cytoplasm. This led to a deeper understanding of cell diversity. Modern light microscopes are used to observe basic structures, such as cell walls, membranes, and chloroplasts, while electron microscopes provide far greater detail, revealing tiny organelles like ribosomes, mitochondria, and even the layers of the cell membrane. These powerful tools have allowed scientists to classify cells not only by what they look like, but also by how they function and how they are organized internally. Scientists often use diagrams to represent and compare different types of cells. Diagrams of prokaryotic and eukaryotic cells show how their internal structures differ. For example, prokaryotes lack a nucleus and organelles, while eukaryotes have clearly defined components. Plant and animal cell diagrams are also used to highlight key differences. Plant cells have a cell wall, chloroplasts, and a large vacuole, whereas animal cells do not. Such diagrams help students and scientists visualize cellular features that cannot be easily seen with the naked eye. When paired with microscopic images, they provide a clear way to classify cells and understand their roles. For instance, diagrams from electron microscopy show how chloroplasts capture light energy, or how mitochondria release energy from food.